Welcome back, everybody. So IITE seminars are back for this semester, and it is my great pleasure to welcome Virginia Domínguez-García, who is talking to us from Seville, and we're going to be learning, I guess, something about how network theory can help us understand persistence in plant-pollinator networks. So without further ado, please, Virginia, the floor is yours.
Brilliant. Well, hello everyone. I would like to start by thanking the organizers for letting me present my work in this seminar series. Um, Today, I will be sharing part of the research that I have conducted during the Marie Curie project that I have been developing at the Doñana Biological Research Station in the southern of Spain. So the main goal of this project was to move forward some more integrated measure of ecological stability that combines both theoretical advances and empirical data. And since the audience here is likely more familiar with the theoretical parts, I will also include a lot of this empirical side in the work that may be more interesting to you, maybe. So without further ado, let's proceed with the first part of the talk. Uh, oh, how's it now? And in the first part, we will see how the structure of the interaction network can help us to explain how the species persist in the field. And this study I have done in collaboration with some colleagues of my current institution. So let me give you some context on why this is important. So it's increasingly evident that we are experiencing rapid global change that is driving the extinction of many species while human populations continue to grow. So these two processes are happening almost in parallel, as we can see here. And these are reshaping ecosystems and challenging biodiversity on an unprecedented scale. So this means that there is an increasing risk of extinction for many species of plants, mammals, and most importantly, for our focus today, that is insects and in particular pollinators. So in such a scenario, as changes, understanding and predicting how ecological communities will respond has become more crucial than ever. And that is precisely the goal of ecological stability study, to understand and, as far as possible, to predict how ecosystems are going to respond to these external changes. However, despite constant growth of ecology, one major challenge remains, and is not less advanced, and that's the need for a deeper mechanistic understanding of how communities function. Basically, in other words, is that we must identify the process that are driving the ecosystem response if we want to know how they are going to behave. So, as probably all of you know, if you have worked at some point with ecological theory, gaining a mechanistic understanding is challenging. We need to develop models that simplify the reality, but still capture the key mechanisms that drive the ecosystem responses. The challenge is that these models of complex systems are by nature also complex themselves and involve in many ways many parameters. So in many cases, this makes the direct empirical testing of these models, if we consider them as face value, like all of them together, extremely difficult, if not impossible, since determining all these parameters experimentally would require an enormous effort, assuming that we could even do it in an experiment. So this problematic has generated that historical division in how ecological stability is studied by theoreticians and by empiricists. And it is especially evident if we look at how stability is quantified. So these two studies revised the publication where ecological stability had been measured somehow. And what both found was that only between 2 and 4% of the studies combined the use of theoretical models with empirical data. So this division may not seem important for you, especially in a heavily theoretical environment like this, but believe me, it's actually dramatic because it makes impossible that the advances made in theory can be translated to empirical systems. It also prevents that the empiricists design experiments to test those theories, and all in all, it difficults the transfer of basic ecological knowledge to conservation planning in the real world. And that makes me, and should make all of you, very sad. However, don't despair, because this talk will be really short, and it's because there is a growing awareness of this problematic. There is increased effort on part of the empirical and of the theoretical community, which are those that I am most aware of, to try to close this gap. In particular, today I will be talking about the theoretical framework that makes it easier to connect theory and empirical data on ecological communities, and that is the framework of the structural stability. So, for those who have never heard this before, these concepts appear for the first time applied to biological systems around the start of the 90s. In general, we say that a system is structurally stable if, when we change its parameters, its dynamical behavior does not change. But what does it mean in ecological systems? So, since despite being a theoretician myself, I was not familiar with this concept until more or less recently, I will explain it with a little detail and give you a glimpse of how this framework works. Okay, so we are dealing with model communities of this type. How do we usually model an ecological community? So if you are probably very familiar, we do that using differential equations that tells you how the population of the species evolved through time as a function of the population of the species and some parameters. In the most simple case, this function is just a constant increase, like in this case. I reproduce proportional to my population. This reproduction rate, this R highlighted in green, is usually a consequence of the environmental factors and will change accordingly to them to reflect how well a given species is doing in our environment, let's say. So when we solve this equation, we obtain the behavior of n through time. Basically, that's what we want to know. How many species, how many animals I'm going to have in one species. So in this case, we just know that the population will grow without limits, basically. So the next thing we can introduce is just a species interacting with itself to include some self-limitation. So now the population depends both on how well the species is doing in the environment, these are in Greek, but also it's limited by a current capacity that is modeling some resource, basically, meaning that n cannot grow indefinitely. In this case, when we integrate this equation, basically we will obtain a curve that will saturate at some point. And that was for one species, but we could consider more than one. First, we can consider them in isolation, like these two. Each one has their own carrying capacity and their own reproductive rate that will depend on the environment, these are. And if we just consider this and we run the simulation, basically, we will obtain that the two populations arrive at some equilibrium. However, we know that the species are not living in isolation, but interacting with each other. So we can model that by introducing these interactions among them. So if we switch these interactions, like for example, let's say that these two species are going to compete, we see that the population of each species is going to be determined now, on one hand, by the environment. These conditions that are highlighted in green, but also by the biotic condition, that is, by how two species are interacting with each other. This information is encoded in these A parameters that constitute what is called the community matrix. So basically it tells you how each pair of species is interacting. So when we switch these interactions, the final population in the steady state change, as we saw. So basically are these population in the steady state that we are interested, and we are going to call them N star, that is population that don't change over time. So it's debatable if nature really works like this around a fixed point, but for the moment, being a theoretical community, let's just assume that it works like this and will continue based on this assumption. So when working with these generalized Lotka-Volterra systems that don't have nonlinear function forms, like a handling time or something like this, because in the steady state, the population remains constant, the derivative of n are 0, and then I can set these derivatives to 0 and solve on the equilibrium the values of these n stars. applied to what we see here, this means that my differential equations now simplify to this linear system of equations, okay? Where I have the reproductive rates in one side and the interactions and final populations, the N star, in the other. Which in a more compact form, in a matrix form, if we had more than 2 species, could be written like this, where A is the matrix containing the pairwise interactions among species, and R, the reproductive rates of the species in isolation that will depend on the environment. Okay. So in this case, which means that by inverting this function, I can obtain my final population, this N star, as a function of both the growing rates, the R, and the biotic condition, this R, okay? Which is basically what I was looking for, the N star. So in this particular case, because the local terra system are easy to deal with two competing species, it's even possible to solve it analytically, such that the equilibrium population of these two species are a function of the model parameters. And we see there, like N1 star is a function of this A's, and N2 star is a function of this A's and R. In general, the local terra system can be solved in this way. But what is more important is that I'm only going to be interested in values of N star that are either 0, meaning the species are extinct, or more important, above 0, because negative populations do not make sense in ecological terms. I cannot have a negative population. So we will say that the community is feasible, and we will be using this for the rest of the day probably, if all species have populations that are above 0. Okay, so my condition of feasibility is that one. And as I said before, since I am interested in communities where all species survive, that is, where all N stars values are above 0, I can write the vector of growth rates, these R's, as a linear combination of the columns of the community matrix like this. So basically, this is just a rewriting of the equation in the left. So now my R vector formed by R1 and R2 is a combination, because N1 star and N2 star are just two numbers of these two vectors of the community matrix. What this means is basically what this figure shows. As long as these N stars are all positive, the vector of R, of the reproductive rates of the species, is going to be confined inside these two directions. So keep this in mind for later, because we will use it. Sorry, it's, I don't know, someone calling. Okay, so going from two interactions to more species is easy. So my model community in this case will look something like this. I have this species where the cycles reflect the population size, the population sizes that are going to be determined by two key factors. Basically, on the first, on the one hand, we're going to have the biotic interactions, here represented by these blue lines that we are going to consider fixed. And on the other hand, we will have also the environment, these R, okay, that will determine also that they reflect the climatic conditions, for example. So when my community is exposed to external changes, as can be, for example, changes in rain patterns or changes in the temperature, if when confronted with that, our community does not change its behavior, then we will say it's structurally stable. While if, on the contrary, there is an important change in the behavior, such as the extinction of some species, we will say that the system is not structurally stable, which means that it's sensitive to changes in its parameter. So that's the general idea. But it wasn't until 2014 that these other authors came up with a way to quantify this structural stability. Because for now, it's only like a concept. So the basic idea behind this is that the network of ecological interactions between species that compose the community, the plants and pollinators in our case, is going to determine the range of environmental conditions in which the species in the community can persist. So remember that we have just seen that the environmental conditions are what determine the reproductive rate. So this diagram here represents on the x-axis the abstract environmental condition for pollinators, and the y-axis the conditions for plants. And the area inside these lines is the joint environmental conditions on plants and pollinators, in which the networks, architectures that are seen there allow all species at the same time in the community to coexist. And the points represent the real estate where each community is. So given the air, basically. So if these communities were to be exposed to changes in the environmental conditions, which in modeling terms we translate in displacing these points that represent the R, the particular R's, we see that depending on how the interactions are organized, the communities will be able to resist more, like this central one that has the green arrow, in which the point is displaced, but it lies still inside the area of tolerated condition. While in the other cases we see, like the one up there in red, a similar displacement can take the community out of the tolerated conditions for this persistent. And with that, we will lose one or more species, and basically the system will not be feasible anymore. So the quantity the authors propose to quantify structural stability is this area of tolerated conditions that allow the persistence of all species in the community. And that's what they call the feasibility domain. So it's worth noting that here one more important thing regarding two concepts here, stability and feasibility. So maybe most of you are already aware of this, but just in case. So going back to our system of two competing species we saw before, the local system has the advantage of having been studied for, I think, 100 years now. That is the birthday, which means that a lot of the analytical knowledge has been developed for them. So in these systems, the condition for the existence of a global equilibrium point that is stable, which basically means that when the system is perturbed, the perturbation will fade and the system will go back to the original state, is that the interspecific competition strength are lower than the interspecific. So here basically is what's written here, that the diagonal terms are larger than the other ones. However, even if that condition is satisfied, depending on the parameterization that we have, we can have different scenarios regarding the species abundance. So we can have, for example, both species surviving, as is the case in A, or one species having negative abundances, as is in B and C there on the right. That is because the feasibility condition is a different one to the stability one. It satisfies if these values, these star, are positive, which eventually can be translated in this case to relative values of R1 to R2, which in this figure, for example, what this means is that the community will be feasible only in case A, where the ratio of R1 to R2 satisfies the feasibility condition and is in the white area. The issue is that many studies have assumed that a feasible equilibrium exists without rigorously verifying it. However, stability results are actually only really meaningful if the equilibrium point that we are trying to assess is feasible, because if not, we are considering the stability of a system that lacks ecological realism. So the coexistence of species, as we see, depends both on stability, whether the system returns to equilibrium after perturbation, and also feasibility, whether such an equilibrium exists under realistic conditions. So different parameter changes can lead to situations where an equilibrium is stable but not feasible, or feasible but not stable. So in this sense, the structural stability quantifies how broad the range of conditions is for a feasible and stable equilibrium to exist. And the approach shifts the focus from merely checking if stability conditions holds to evaluating how robust a species coexisting is under varying ecological parameters. So going back to what we were seeing, to quantify this area of parameter space compatible with species coexistence is into this pretty easy. One just needs to consider that angle between these two vectors, and the more or the larger that angle, the more structurally stable that my system is, as the vector R, this black vector, let's say, can change more and can accommodate more changes without leaving the feasibility region. Going to 3 dimensions is a little more complicated. Something like this. You have this solid angle with the 3 directions, and as long as you remain inside, you will be feasible. So further theoretical advances, like ones we show here, have allowed us to compute the feasibility domain of a generalized local Terra system in an analytical way for a system of any size, basically. And this quantity, basically the solid angle, compresses inside this direction. That is here shown just for curiosity. However, one thing about this form is that this form of measuring has the problematic of decreasing with system size. I move this. Decreasing the system dimension, because as the number of dimensions increase, this quantity becomes smaller and smaller in proportion, which means that you cannot really use this, large omega to compare systems of different sizes. To do that, you actually have to use the normalized version, which is this small omega, that we can interpret as the average probability that when I choose a species, it is persistent. Both quantities, these 2 omegas, are a function of the community matrix A that encompasses the biotic interactions, which means that in this framework, the structure of the network will determine this omega, and with that, the possibility for the species to persist. Okay. So far, I hope I have transmitted to you the basic idea of what it is and how can we quantify structural stability. But it is possible to put it to the test on empirical communities. So that's precisely what we decided to look into this work. What do we need to do that? So basically, we need 2 things. On one side, we need all the theoretical framework that we have just seen, in which we will use local Terra type dynamics to model the species populations in this fashion, where we are modeling just the final equilibrium state. So these models basically compute the population of a species in a fixed point as a function of their reproductive rate, as we see, and the interaction competitions that we will consider as mean field, competition within the species in the same group, like plants or pollinators, and also the mutualistic interaction, this gamma, between plants and pollinators. On the other hand, we will also need the field observations in our community, that is, the empirical data. In particular, we are interested in persistence, since it is what we are going to study, basically, the persistence of a species as a function of the interaction network. So, I'll detail the empirical procedure a little bit, because coming from physics, I was kind of impressed when I realized the amount of work that was needed to obtain this type of empirical data. So for this study, we have used a database of 12 sites that are located in the south of Spain, more precisely around Doñana, as you can see in that map, that have been sampled for 6 years in a row. I mean, now more, but when we did this, 6. Each year, we sampled twice a month for at least 7 rounds during the full flowering season, that is from February to June, across these 12 sites. And these are located in a landscape fragmentation gradient, okay? And we gather data both on flower abundance and plant-pollinator interactions frequency. So these sites were at least 3 kilometers apart, which means that, which is larger than the foraging distance of pollinators, which means that they can be considered as independent. Okay, so these sites are all open forest island, like this one you see here, which has some trees, and most are just grass and open terrain. So in each site and during each round, basically, the people that knows the species walk 100 meters straight line for 30 minutes, in which they wrote down every plant-pollinator interaction seen, differentiating between the number of pollinator individuals and the number of visits per individual, such that then we could retrieve the abundance of pollinators. We also measure the abundance of plants by recording the number of flowers of each plant species in 10 1 meter square subplots, along this transit, like this that we are showing there. So, so this intensive fieldwork resulted in a dataset that contains more than 170 pollinators and more than 30 plant species. Basically, those that we are seeing there. So, aggregating across all the year, we can know which species has been present each year in the field. And finally, we quantify the empirical persistence of the species as the number of years a species has been present in the field, as we see there. So basically, for each species, the empirical persistence is how many years of those 6 it has been present in the community. However, remember that apart from the persistence, we were also interested in the empirical network of interaction, since, as we have seen, interaction networks seem to play a fundamental role in determining structural stability. What we will do is introduce these empirical networks inside our mutualistic model. And we will have so in that case, we'll have a model with a real structure, a skeleton, and we will run a generalized local Terra dynamic. So a little detail on how to construct that networks models. So in these same 12 sites, at the same time that the species abundance were recorded, interaction between plants and pollinators were also recorded while walking these transects. So this added up more than 250 hours of observation in the field and resulted in more than 1,400 interactions. Since we have various years, basically we constructed the network of all possible interactions by aggregating through all years the interactions at each site. So this meta-web, which we have one for each of the 12 sites, reflects the potential interactions in each site. And we will consider that a pollinator and a plant are connected. That is, this gamma is not 0 if they have interacted at least once. And they won't be connected, gamma will be 0, if they have never interacted together. So introducing this network in the model that we saw before, basically this was the great omega, the great gamma, we can obtain the effective biotic interaction between a species in the same trophic level. That is the interaction between plants and the interaction between pollinators, here represented by these links between the species in the same level. So we consider mean field interacting competition, as we said before, and that will be modified by this mutualistic interaction. So with this with these networks, basically, or with this interaction matrix for pollinators and for plants, we can then compute these small omegas with the procedure that we saw before. Okay, so for each network, we will have this small omega that will tell us basically how easily pollinators are expected to persist in the field. So then, applying, as I say, the method that we see before to quantify the structural stability, we can obtain these omegas, and basically they represent the probability that when I choose a random species in a community, that species is persistent. So basically, if we look at this, basically the hypothesis that we are going to test is that if we have seen how we can make it, if we have 2 different sites and we obtain from them the empirical mutualistic network, with this, we can obtain the effective interacting matrices. And using these alphas, we can compute the expected probability. So basically, the interaction network theoretically determines how easily the species in the community can persist. For example, in this case, in site A, the chosen species can change its reproductive parameter a lot, along this green axis. While inside B, the species can only decrease its reproductive rate until a moment that is less than in the case above. So basically, it is easier for that species to disappear in site B than in site A. So we expect that these differences generated by the interaction networks are visible in the field when we measure the persistence of the species. That is, that sites where the network has higher structural stability, we will see also, or we expect to see more empirical persistence. So to see if that happens or not, what we did was to correlate the expected persistence provided by the model, here represented in the y-axis, with the empirical persistence of the community. That is, how many years in average are a species present in the community. And what we see is that there is a high correlation between the expected persistence by the model and the observed persistence in the field, the empirical, at the level of the whole community at least. So this means that the sites with which the model predicts that in average species are doing better, we see that they are more persistent in the field, that they are actually doing better. We also did it at the species scale, being very fast. Basically, and now we will consider the probability of the species to exist in the community. So in my case, in this, if we have 3 species, the probability of species 1 to be there is the combine of having a species 1 alone, species 1 and 2, species 1 and 3, and species 1, 2, and 3. So if we apply this workbench and we do it at the species scale, we see that the positive relationship, in that case, what we will represent is the probability of persistence for each of the species in its site against how many years that given species has persisted in the field. So what we see is that basically we still see a positive correlation. And in these sites, let me explain that all sites are plotted together to save space, but we repeated the same analysis independently by site. That is, we did a Spearman correlation for each of the 12 sites. And that's what is shown there, this row that goes from 0.37 in the worst case to 0.65. All of them being statistically significant. So this means that exists a positive relationship in all species independently, and that pollinators that appear more years in the field have high persistent probabilities also according to the model. Finally, although it does not appear here, the figure, but since we know that ecological stability has different phases, what we decided to look also was temporal stability, like how much might the biomass oscillates in time. That's the inverse of the coefficient of variation. However, we did not find any relationship at the community level, which means that being persistent in this case seems to have nothing to do with having more stable populations. And when we look at the species scale, which is this figure in the lower part, we found that in the x-axis, we see either the pollinator expected persistence, this omega, or the real empirical pollinator persistence, and in the y-axis, the temporal stability, that is the inverse of the coefficient of variation. And we find a slight positive correlation, but basically it's induced because species that sustain stable populations are the more persistent. However, as we see, persistent species can be either stable or not. I mean, they have the two, they can do pretty much everything. Okay, so now that we have seen that the network of interactions can determine the ability of pollinators to persist in the field, we went on asking ourselves to what extent did landscape affected pollinator persistence and how. So first of all, let's take a little step back and see why I'm talking about landscape and pollinators. So in general, there are pollinator declines that are more or less intense depending on what we are looking at, but they are well documented. And there is more or less an established consensus on what are the most important drivers of the drivers of global change that are behind pollinators decline, and that we can see here. So they are land use change, land management. So if there is intensive agriculture practice, for example, using herbicides or pesticides, of course, also climate change that can alter the phenologies of plants and beings, and also invasive species. But among the most important drivers are land use change with the consequent loss and fragmentation of habitats. So there is ample evidence with many reviews and synthesis on the matter pointing to the negative effects that habitat loss and fragmentation has, and intensive agriculture has on bee abundance and richness. Even if in some studies there is a slight improvement of pollinator in urban areas, but this mostly can be because there are like some parks or some pollinators even that have been introduced in the city to help pollinators. But in general, the more intensified and the smaller the patches, the worse for the pollinators. However, while the negative effects of flankers on pollinator and pollinator richness are proven, it is not clear how the process operates. And in general, basically, landscape intensification can affect bees in two ways. First, directly, for example, by reducing the nesting site, making it more difficult for bees to reproduce, or because the proximity of pesticides can directly harm them. But there is also the possibility that landscape also determines the persistence of pollinators by changing the network of interactions. For example, because there are less plants or less plant species, less abundance, reducing the visitation frequency. And also, there is like a lack of agreement on how landscape changes affect the network of pollinators. So in light of this, we ask ourselves, how does landscape affect pollinators? By directly affecting their ability to function, or by indirectly introducing changes in how the pollinators interact. So if you remember, our sites were distributed along a landscape fragmentation gradient. So we will try to study this here. So what we are going to do is that on one hand, we are going to track landscape. So we mapped the different uses of land in our sites in 1 kilometer radius from the center and quantify the percentage of cultivated land. And additionally, we also measure patch size. So I must say that since this study was not originally designed to study the effect of landscape, we find that the relevant variables are almost all correlated. That is, we find that in general, larger patches are those with more natural habitat, for example. On the other hand, as just showed before, we have just seen that the network of interaction is playing an important role. So we are going to quantify its structure using some network level metrics, such as nestedness or modularity. And remember that we also have the persistence of species in the field. Okay, so when we looked first, was the effect of landscape fragmentation on the average expected probability persistence. So this figure, the lower left part, shows in the x-axis the patch size in logarithmic scale, and in the y-axis, the average persistence, this omega. And we see that communities with more expected persistence, larger values, tend to be those situated in larger patches. Okay. So with a Spearman correlation of 0.6, which is marginally significant. And in general, they also tend to have a more cohesive structure, or what is the same, a nested structure that we'll see later in more detail. This dependency with patch size can be important because this means that landscape fragmentation can make ecological communities less structurally stable, which in turn means that they can be less able to withstand environmental changes. Basically, then we examine other structures we search the structure that most determine the species persistence in the field, both the theoretical and empirical. And what we found was that, well, nestedness was positively correlated, modularity was negatively, because these two in this case are countercorrelated. But and what a nested structure means is that if we order the interaction matrix by decreasing degree, we find these nested patterns which maximize the number of shared partners between each pair of species. So anyway, in this case, what we found was that the metric that most correlated with the pollinator persistence was the number of shared networks. So the more mutualistic networks that you share, the more pollinator persistence that you find in a given community. Then, in order to see to what extent the landscape has a direct effect on pollinators, or if it's mainly through a reorganization of the interaction, we perform a path analysis, connecting on one side the different metrics of landscape, that is, percentage of cultivated land or patch size, and on the other hand, the interaction network structures, basically the number of mutualistic partners or the nestedness, and finally, the empirical pollinator persistence. And what we found was that landscape seems to be determining the pollinator persistence mainly indirectly through reorganization of the network of interactions. However, finding how landscape determines the network interactions is not trivial, and we are still working on this. And for now, what we have seen is that just the larger patches are are more cohesive, more nested, and have more shared partners. That's not obvious why that happens. Okay, so now let's finish with the second part. That is also part of my Marie Curie project. In this one, we are looking at how networks behave in time. So I hope I convince you of the relevance of the network of interactions of the pollination studies in order for them to persist in the field. However, when we go to the field to get those networks, usually we don't go one, we go two, and if we are lucky and have enough money, more times, like 3 or more. So in our case, we have a long-term experiment where the fields have been observed for 8 years. And if you remember from what we saw before, what we did in order to obtain the most complete plant pollinator networks was to aggregate over all years in order to get the potential network of interactions. So the thing is that if we look in detail at each of these annual networks, they are different from one another. That is, the network we saw the first year is not the same that the network we saw the second year or the third year. However, in order to get this detail, you need to repeatedly sample one site, which is not too common. This means that what we usually have are one snapshot of the network of interactions, not the whole picture or the different snapshots. This type of long-term observation that allows us to study the changes in plant pollinator networks through time are slowly increasing because people is growing more aware of the importance of this temporal dynamic. And this increased resolution, however, comes with a cost because now we don't have only one interaction network. We have to characterize a lot of them. And there is already a good body of literature characterizing these temporal plant pollinator networks. And what so far is known from studies evaluating the annual turnover is that, in fact, the plant pollinator communities are extremely dynamic, with more than 70% of these interactions occurring only once. This increase in resolution, and there is, and also, yeah, and also these intense changes mean that the species identity is inherently variable within networks. The general consensus is that there are conserved structures at the community level, as are, for example, the nestedness or the core periphery structure. However, it's yet unknown the ecological consequences of this interaction reorganization for the maintenance of the species diversity. It has long been suggested that temporal reorganization of plant pollinator networks may promote greater pollinator persistence, but these hypotheses have never been put to a test, and this is what we will do here. So basically, what we want to know is how those interactions happening in the field affect the ecological community, and in particular, its persistence. So I will go rapidly through this in order to do what is going to be basically one useful way to characterize interaction turnover is to take the idea of quantifying the beta diversity of interactions. So in this study, the authors propose a way to do so by comparing across pairs of networks how many of the interaction changes were consequence of a species turnover and how many were consequences of rewiring. However, what we need to do is less difficult because we only need to deal with one dimension, which is time, which means that we are going to compare our networks in time t with networks at time t plus 1. And then we would like to know if the changes of these interactions turnover are because of a species, because of rewiring, like this, where all the species remain but some interaction changes, or if they are because of a species turnover, because the species changes. So considering this, the first step is that we are going to quantify the reorganization of the interactions, looking at pairs of networks like this. So this will allow us to identify which interactions happens, comparing networks at time t and t plus 1. We will identify first the species that are permanent during these 2 years. Here we see them because they are in dark green. And we will also see the species that are only present in the first year in blue and in the second year in yellow. This will allow us to identify the species that happen, the interaction that happens in both year, also in dark green, and the interaction that happens only in the first in blue, or in the second year. Note that this matrix shows simultaneously the interaction from the year before and the year after. So basically, using these annual empirical networks, we will assess the expected average survival probability. That is this empirical omega. And for each site and year, we will have an average survival probability, empirical survival probability. As we are interested in knowing the relevance of this organization for the functioning, we will compare the structure with different randomized counterparts obtained with 3 null models. So using these different null models, we will create randomization of the linear empirical networks. And the basic one, it's weird. Sorry, there is an animation. So basically, what we want, no. So then we will quantify the expected survival probability for the pollinators in these synthetic communities using the same workframe that we have used before. And and what we're interested is in knowing if the empirical interaction reorganization is somehow helping the community to survive. That is, if the opportunities of survival are greater in their empirical networks. So the brief description of the null models. So this represents the network at year 2 with the permanent species colored in green and the new or transient species, those that are not present in the previous year, in yellow. And we can divide the matrix in 4 blocks according to the role of the species that interact. Permanent with permanent plants with transient pollinators, transient plants with permanent pollinators, and transient plants with transient pollinators. So we created 3 different null models. In the first one, that we named random, we allow all non-permanent interactions to change, keeping the number of links inside each of these 4 groups that we saw. So we reshuffle all the yellow links, as we see below. In the order to see the role of the permanent and transient species separately, we devised other 2 null models. In the second one, that we named random rewiring, we reshuffle the transient interactions among the permanent species, those that are marked in red. And in the third null model, that we name random turnover, we reshuffle the transient interactions that come from the transient species. In this way, we are able to distinguish the effect of interaction turnover, but also the interaction turnover caused by rewiring or by species turnover, following the philosophy of these previous studies. So here, our hypothesis is going to be that if our empirical networks are somehow better, we should be able to see them in this modeling framework that we have seen. That is, that the empirical organization may be helping, if empirical organization is helping a species to persist, we should be able to see this using this structuralist framework. Okay, so what we found was, first, looking how network changes, we also found that our pollinator plants communities were highly dynamic, with most of the interactions, 90% being present only once across all the years. So basically, as you can see here, the average number of pollinators is smaller than the average number of permanent pollinators is smaller than the average number of transient pollinators, approximately by a third. However, this is not the case for plants. And in this case, slightly more plants are permanent, and the ratio of permanent to non-permanent is half-half. We also find that the interaction changes are distributed like this. Basically, most of the interaction changes happen between permanent and transient species. That's roughly 60%, while 20% happen between permanent, that is, are rewiring, and another 20% are because changes between 2 transient species. So, interestingly, when we look at how interactions are distributed between permanent and transient, we saw that most interactions are concentrated in the core formed by the permanent species. We support the prior evidence that generally species tend to occupy central roles. So then, to see how these reorganizations affect persistence, what we did was, for each site and year, we use a null model, 3 null models, to generate 500 randomizations of the empirical network and quantify the average persistence, this omega. And this is what is shown here. For one particular site, for each year, the distribution of these omegas of the synthetic randomized networks. The empirical value is the average persistent probability is represented by a dashed vertical black line. So to test whether the observed interaction to over in plant-pollinator networks promote greater persistence than expected by chance, we compare the empirical community persistence with the obtaining by these known models using zeta score. So to quantify, basically we are going to measure how far is the average of a given null model from the expected empirical average. So we did this for the 12 sites for the 8 years, and that's the distribution I'm showing here. The distribution of the zeta scores for the annual networks in the different sites. And the distribution is what our analysis revealed that the empirical network structure observed in the field generally enhances species persistence because most of the values are above 1. So the zeta score distribution of omega peaked around 1 for the random and random rewiring models, with majority of cases being above 1, but in contrast, the random to over, the majority of networks have zeta scores below 1, indicating that the server networks tend to underperform randomized control. So basically, our results indicate that the process of interaction turnover, especially rewiring, is promoted greater persistence than expected by chance. However, if we look at these green vertical lines, that they represent the value of the 1% of the better cases of the randomization, we see that the empirical networks are not optimal. In some cases, there are some synthetic configurations that are the better than what we see in the field. And lastly, basically given the critical role of rewiring among permanent species, we delve deeper into these interaction changes, and to better understand the underlying drivers, we explore to what extent the observed interaction changes are due to either changes in species abundances or in their changes in their phenology, because of previous studies suggest these are important. And basically that's what we did. Here in the x-axis, we represent the changes in the abundances of permanent species and changes in the phenology, phenophase, like how many days it changes for permanent species. And we see that the linear correlation indicates that the amount of rewiring is significantly proportional to annual changes in the abundance and in the phenology of the permanent species. So the larger total changes in abundance and in activity of period, the more rewiring the network exists. And finally, we also found that more changes caused by species turnover go hand in hand with an increased rewiring among the persistent species. We suggest that the addition or loss of species that not merely alters the network, but also indirectly reshuffles, reshaping who interacts with one, which has been previously shown in experimental studies. So, and that's it. I will, because I take too much time, I will just go with the last one. And if you want, we can, you can ask me a question. But this are like the main take-home message.
Thank you very much, Virginia, for the great talk. So, if I understand correctly, we can open it up for questions. So, if you have questions,
yeah, of course,
please just raise your hand in the participant list, and I will call on you. So, are there any questions? Yes, Chris, go ahead.
Hey, thanks so much. I was curious to see that the nestedness and modularity and those things did a better job of predicting persistence than the structural stability metrics that you had. In terms of the correlation was higher at the very least. So I'm wondering a little bit kind of what the role of the structural stability metric in this is. Because obviously it's going to be super hard to estimate it accurately because you're essentially taking a binary matrix, inverting it, and getting out the omega. So I'm just wondering kind of how you see the extra added value of structural stability in that case when other measures seem to do a better job.
Yeah. So that's true that if the structure gives you everything, maybe it can be a shortcut. So in this case, we did basically we did a very, how do you say, thorough search of the parameters to see that these correlations remain, and basically it sustains. So I guess that there is a real relationship behind, which means that how the species are connected is directly affecting of the ability of a species to persist, because we know that this omega is a function of A, so it makes sense that they are correlated. Maybe it's enough with knowing the structure. I mean, that's somehow is the underlying message of all of this, that somehow if you find a metric that really is able to predict the community behavior, maybe you can use it. Still, I wouldn't say that these are completely how do you say, interchangeable, because if you can always change parameters in the dynamical models, and you can do some, how do you say, you can touch them a little bit and still find some accommodation. So I wouldn't go so far as to say, throw it away, only consider structure, because I haven't done enough tests like to throw away all the dynamic still. But I guess it's worth considering how these structures are maximizing the species' probability to survive in the field. So I think there's still more work there. So I wouldn't go as far as to say, forget about the model, just look at the structure. So I need to know more, but it's a good point.
Thank you very much. Are there any other questions? People are thinking maybe I will ask a question. So you might have actually touched on this, in which case my apologies, but any sort of feasibility domain calculation will critically depend on the diagonals of the matrix. So how are the diagonals estimated?
Yeah. So in our case, the diagonals we use the typical model where you put the diagonals to one, and then you put the average, how do you say, the mean field competition to a given value that is rho, that you then can basically tan, and usually you have to put it to be less than one, because if not, everything will kind of die. And then this mean field competition is modified by the mutualistic interaction. So here, maybe I didn't.
Yeah, I remember, but you should go back anyway. Yes, there it was, I think.
Yeah. So here, basically, we have these two. So in these alphas, basically these are blocks where the diagonals of cell regulation is one, and then the competition within guilds is set to a fixed value. So in the end, basically by playing with the mean field competition and the value of mutualistic gain, you always, you can obtain a range of results where, in general, the positive correlation that we see remains, as long as we are within a mutualistic region.
Okay, so sorry, the reason I'm confused, I'm just trying to put it together. How so if the alpha matrix is fixed, is the mutualism matrix somehow scalable? Or do you keep it scalable? Or does your measurement method sort of fix its value?
Okay. No, so basically for the moment, what we use as a mutualistic network, which is here.
Yeah, I think it's easiest to have that matrix on screen that you just had a moment ago, right?
What?
Didn't you have a matrix?
Yeah, sorry,
There we
because
go.
I have
Perfect.
the
Yes.
two, so I don't know which one I'm.
That's the one. So
Yeah,
you say that the C matrix is essentially a fixed one with a one on the diagonal
yeah,
every
yeah,
unit and a guild of diagonal mean field value.
yeah, yes.
But the gamma matrix then is does that arise? Is that fixed down by the way you estimate it? It-
So
no,
the
yeah, sorry.
sorry,
Uh,
finish the question.
I was just wondering, is there a constant multiplier as a degree of freedom or parameter that you keep? I'm just asking because the relative magnitude of C and gamma is very important, I assume. And I also assume you have no way of knowing their relative magnitudes in reality.
Yeah, yeah. So what we did here is that we use to parameterize this gamma matrix. We consider the empirical as a skeleton, the empirical interaction without weights. So if two species interact, we put a one. Then the whole matrix, basically this gamma p and gamma a are these one and zeros multiplied by a parameter. This is gamma zero. It's as you said. So basically, it's the interplay between the mean field competition that we consider and this gamma zero, that is the mutualistic gain. What is going to decide how strong is competition with respect to mutualism in here? So because we cannot know with our empirical setup the how do you say the relative importance of those two, what we did was like a parameter sweep. So we kind of search of all the results how far you can go when strengthening competition and strengthening mutualism, and still have some result that is consistent. So basically, what we see is that as long as the competition and the mutualism are kind of uh, strong enough itself, we see this positive correlation. Of course, to the best of my knowledge, with the data we have, we cannot empirically determine which the importance of competition versus mutualistic gain in the field. There are other ways and other communities where this is more doable, uh, and that I'm trying to work with them now. With plants, for example. So in that case, you can maybe, if you know the network of real competition and then the real network of mutualism, then you can better have a better idea of what's happening. But for that, I think you need to like build the setup, the experimental setup or the
Yeah,
observation
no,
field.
and of
Here,
course that's a huge undertaking. That there's no question. I'm more wondering that in this context, you say you swept through the gamma zero, which is a great idea, of course. But what I'm trying to wrap my head around is what determined which gamma zero values were more plausible or better than other gamma zero values.
yeah, what I did was to use the same values that they have been used before in the when they presented the model that I used, basically that they have used for other empirical uh, system. So basically, they did something similar as I did before, and they used this mathematical modeling, and I kind of use as a baseline the parameters they use to how do you say to model their empirical communities. So that was my baseline, and then I kind of do the sweep just to be sure of the robustness of the results because in the end we have a lot of parameters to go through. Basically, the interplay depends on how strong is the competition with regard to mutualistic benefits in the end.
Okay.
So as long as they are, let's say, in a similar range as the ones that they propose, everything uh, is the same. as long as you start not having mutualistic benefit, then you don't see this correlation, because of course you only have the competition, and then you are not including the network of mutualistic interactions. So then you stop seeing this correlation with the structural stability and the field empirical results, because it's like not putting any information in the matrix at all. So you basically you won't only see everything of only competition, basically, and no like random among them.
Okay,
So you have to basically you have to have a mutualistic benefit that is strong enough that it shows in the model, basically. If that's, I don't know if that's a good answer, but in modeling term, I think it's the best I have.
Okay. No, I think I have a better idea now as for what was going on. So thank you. No, that yes. Are there any other questions? There are no more questions. Thank you again for the talk and for coming.
I think Chris had a question, but I'm not sure.
Only to fill if people wanted. I was slightly curious about the you compare your different sites with quite radically different sizes, but the number of species in each wasn't that different. So I just wondered, kind of, is that the number of species you see along the transects, or is it the number of species in the sites, or kind of how that worked through?
Sorry? So the number of species,
You had a little table where you had the size of the different sites and how many species
yeah,
you saw. And
yeah, I think was before that one.
yeah, and I was just
Yeah, so
surprised at how it's really quite similar numbers, just by radically different sizes. And so I just wondered kind of how what do you think that might be explaining in there? Or
yeah, so I'm not sure, but I basically uh, what this is showing is the number of species that has been detected in each site. So I think that even the smaller sites are similar to those that are larger. And actually, we don't see any how do you say trend of richness of plants or pollinators with the patch size, which was kind of surprising, but there is not. It's like probably what happens is that in the smaller patches, that one cool thing that they have less richness, they are less diluted, or let's say the otherwise. In larger patches, the species are more spread because they have more places to go. So in the end, you don't see a lot of difference between the small and the large. I don't know if it's that, or if it's just that they are really similar. It's not like they have huge differences among them, because it's the same habitat type. Is this how do you say pine island forest? They are all the same type of habitat. There is no major disturbances, so it makes sense that they are pretty similar. The only important thing here is that they are independent, and they are more used as replicates than as a real landscape change study, because there are not a lot of difference among them. Still, you can see some tendencies, but that there is not shown in the richness of the species. This being said, we also did like a coverage, how do you say, a coverage study of the sites, and all of them were like over 80% of coverage in the species, but like by studying the distribution of abundances. So it's not that some sites are less covered than others. This should not be an issue because we check for that. And all of them have a good coverage in terms of species distributions. Abundance.
yeah, it's just really interesting to see that it pops up in some of the structural bits, the size effect, but it doesn't pop up in the diversity. And it's just, yeah, interesting.
Mm-hmm.
Thank you.
I wonder what the species area relationship people would be saying. But anyway, I'll leave that to them.
I think there is no relationship, I guess.
That anyway, are there any more questions? If not, then thank you again, Virginia, and thank you everybody else for coming. Next talk will be in 2 weeks. Hal Caswell is coming, none other. So we will be in touch and we hope to see you all next time around. So thank you and have an excellent rest of your day. Bye, everybody.